As you know, extending the human health span is a global priority. We have more people that are above 70 in the blue line there. They're becoming more abundant compared to people who are below the age of 70. And we also have what we call a health span lifespan gap. Here you see that average global lifespan is about 73 years, but the healthy part is shorter than that. So we have this gap here where people are still alive, but they're either frail or ill. So what we want to do is and that's what we call the health span. What we want to do is find a way to keep these people healthier and productive, and it's very important because of the shifting demographics. Aging is a universal process, and it's accompanied by age-related diseases. If we could slow down aging, we should be able to increase health and frailty, or decrease frailty, but also combat all the age-related diseases all at once. So without doing a single experiment, but just looking around and knowing about evolution, you can infer that the lifespan, the rate of aging, is actually plastic and subject to change. So the rate of aging varies between the species, as you know. And that means that gene changes during evolution had to change the rate of aging dramatically, really dramatically. Therefore, there have to be genes that set the rate of aging. So what are they? Well, in the 1990s, my lab set out to look for these genes using a very short-lived little roundworm. And we were able to, just by looking for gene changes that extend lifespan, find a gene change that could double the lifespan and keep the animal, importantly, healthier and resilient much longer than normal. Now, how is this possible? It turns out that within animals, and people also, I think, there are a lot of genes that can make you more resilient. And they do so in very different ways. They can enhance cellular quality control, like they can help proteins fold better. They can help the cells recycle damaged material. They can increase the repair of damage. They can improve energy generation. And they can improve the surroundings of the tissues. And they're all here in the nucleus where the DNA is. They're all here in the DNA. Okay, now it turns out, amazingly and surprisingly, that whole batteries of these genes can be switched on at once by master control genes up here, or control proteins. So there are sensors that convey to cells information about the environment. They sense threats. They might sense nutrient limitation, toxic compounds, radiation, etc., in their environments. And they signal to the inside of the cells to activate this resilience battery. So the whole thing comes on all at once. And the gene changes that we made that doubled the lifespan of these little animals were in a gene that encodes this protein right here, which is the hormone receptor. So there are hormones, even in these little worms, that are similar to our hormones called insulin and IGF-1, whose normal function is to promote nutrient uptake and growth. And when you make the cells a little bit resistant to these hormones, they interpret that, anthropomorphically speaking, as a danger signal. So they roll out the resiliency programs. They become much more resistant to all sorts of stresses in the environment, but they also live longer. And they're resistant to age-related diseases as well, a whole variety of them. So here are some examples. Here's our little worm. Okay, so here's the lifespan curve. At the end of a month, all the normal worms are dead in green, but these ones stay alive much longer and they're healthy. The same genes can be changed in other organisms, including mice. Here's an example of a long-lived mouse here. Again, it stays young and disease-free much longer. It really globally kind of healthier. Here's a really nice example in mice. Another one of these nutrient sensors is called TOR, and you can inhibit TOR by changing the TOR gene or by giving a drug called rapamycin. And these are mice that have been treated with rapamycin, different doses, a low dose, a higher dose, and an even higher dose here in white. And you can see that they live quite a long time, more than they would normally live. And again, they're healthier. And you can even get these effects, importantly, if you deliver the drug when the mice are already pretty old. Okay, so these are ways of extending health span. And what these gene changes do, actually, is they kind of reprogram the body, maybe the way evolution has done it, so that it's more resilient from a very early time. And the mortality rate throughout the lifespan is lower. You can also look for ways of slowing aging by watching processes that change with age. Things happen as you get older, and some of the things that happen to your body are bad for you. And if you change those, at least in animals, you can slow down aging and extend the health span. So I'm going to give you some nice examples of these. First of all, the composition of your blood changes with age. If you take a young and an old mouse and you connect them surgically, then their circulatory systems join, and the old mouse becomes younger, and the young mouse becomes older, interestingly. There are cells that become more abundant in your body called senescent cells. They're actually generated by damage, actually, and they become senescent, and it's a certain cell type. And these senescent cells are very inflammatory, so they're really bad for you. And there are ways of clearing them. And if you clear them in mice, it's really quite amazing. The lifespan is not increased very much, but the health span is improved remarkably, and so is the disease resistance, really much across the board. There are certain metabolites whose levels change with age, for example, this one called NAD here. And in at least some cases, if you correct those changes, then by raising them up again, in mice, there are health span benefits. Autophagy means auto, self, phagy, eating, auto-eating, eating yourself. That's what the scientific name for a recycling system that is active in all your cells that can turn over damaged material and make new material. Autophagy wanes with age, but if you raise the level, but with an intervention, mice can live longer, and importantly, they have a better health span, and they are resistant to various diseases. Inside of you, in your gut, you have what we call the microbiome, which is thousands, millions, trillions of tiny microbes that are really part of you and part of your physiology. And the microbiome is important for your well-being, and it changes with age. And in animals, if you correct those age-dependent changes, in some species, it's been shown that you can extend lifespan and health span, and in others, really remarkable effects on physiology improvements. So I think by playing with the microbiome, there's a lot of potential. As you know, famously, the ends of your chromosomes, the telomeres, shorten with age. There are at least 2 studies in mice that have been published showing that if you prevent that shortening, you can extend the lifespan and the health span of a mouse. So all of these interventions that I've told you about slow the rate of aging, and they increase the health span. Okay? And you can do it many, many different ways, which is really exciting. Because we don't know if any of these will work in humans, but we sure have a lot of shots on goal, many shots. Recently, a whole new way of thinking about aging has appeared, and I'll tell you about it. It's called cellular rejuvenation reprogramming. In other words, becoming younger. Now, actually, we've been doing this for a long time. Not intellectually, we have no idea what we're doing, but our bodies know perfectly well, because when older parents have a child, the child is young. There's no age. Age is gone. It's young again. So our bodies know how to do rejuvenation. And you all know from the famous experiments with Dolly the sheep, and then that you can take old cells, and you can produce young animals from them, okay? Or, for example, more explicitly, you can take old skin cells, for example, and you can treat them with some of these DNA regulatory proteins called Yamanaka factors, particular ones, and you can make them embryonic cells where age is erased. It's all gone. So we don't necessarily want to become embryos again. I don't. But it turns out that, amazingly, that if you just, instead of giving these in a very long-term way, you just give little pulses of these factors to animals, you apparently can turn back the clock a little bit. So you're still the same animal, but you're younger. Okay, and this changes biomarkers of aging, it changes the cell's physiology, it changes the organs, and at least in some cases, it can change the lifespan. So up here, you see, this is a mouse, actually, that it's a mutant mouse, has a gene change that makes it get progeria, so it ages quickly. So here's the normal lifespan of the mouse here in blue. If you take these mice, and you give them pulses of these little Yamanaka factors, OSKM, they live longer. The whole entire mouse lives longer. Here's an experiment showing that these little pulses can improve resistance to metabolic disease, insulin sensitivity. If you take an adult animal and you cut the neurons in the optic neurons, for example, in the eye, they don't grow back. But if you do that and then you give them these little pulses, they grow back. And not only that, there are reports that saying that you can so mice get blind when they're very old. Apparently, these little pulses can actually restore their sight. So it's quite remarkable. We don't know. We don't know if it'll ever be safe in humans, ever really be possible to do in humans, but scientists are all over this because there's a very interesting possibility for our health. Even just making an old organ younger or old immune cells younger, that would be really, really terrific. Okay, so as you might imagine, scientists are super excited about this. Aging, you would think it would be the most boring thing in the world, like entropy. Really, entropy? But it is really interesting because there are regulatory networks and systems that control the rate at which it happens, and that's really interesting. And so there's a lot of effort now in more than 100 startup companies, pharmaceutical companies, and academic labs to harness some of these, to learn more, and also harness some of these, this knowledge to improve human health. But I want to make a point that I think is kind of overlooked, and it's really important. And it really, I think, interfaces nicely with the talk that you just heard. We may already have solutions at hand. I already told you about all sorts of ways that you can increase the health span of mice. There are a lot of ways to intervene already using compounds that are either natural compounds or that are off patent, that are readily available, cheap, not that hard to make. And people are taking them. I go to meetings sometimes and some people will come up to me and say, Cynthia, look, I'm taking these 10 compounds. Is that the right combination? And I tell them, it's really sad. I say, first of all, I don't take any of them, which is irrelevant. I could be wrong, but I don't. But there's a reason for that, and that's because I don't actually know if they're safe and if they're effective. That's the first sad thing. And the second sad thing is I will never know unless we change policy, because there's no way to carry out a very large, rigorous clinical trial that will tell us whether they're safe. And the reason for that is they're really cheap and easy to, if we knew that, we could give them to people, poor people all over the world. It would be terrific. Make them more resilient, disease resistant, increase their health spans. It would be great. But they're cheap, so there's no way of, it costs millions and millions of dollars to do these trials, and you have to get your money back. And you can't do that with these kinds of things. So what we really need is a new framework. We need kind of a World Health Organization or International Monetary Fund or something like that, where governments and maybe nonprofits and maybe philanthropists get together, team up, and just start testing these kinds of compounds in rigorous, large-scale clinical trials for effects on health span. It would be like you just turn the crank and you test this one, and then you test the next one, and the next one. We don't know if any of them will work, but you find out. And when you get some that work, and I think you will, that would be fabulous. Then you have to just disseminate them, and that will happen. So I just want to leave you with that as a possible action item for healthier lives on a healthy planet. Thank you.
So we're going to take some questions. So anybody with a question?
Thank you very much. I'm Nino Kinsley. I'm a public health scientist, so I challenge you with a public health-oriented question. We saw yesterday the bubbles, what happened in the world in the last 2 decades. centuries, life has extended all over the place, and we know why the differences are across the world and even within cities. We have a difference in lifespan, for example, in the city of Bern or Basel, between the wealthiest and the poorest neighborhoods of more than 5 years difference. And it's also a health span that is different. How do you integrate your science into action when we know that the very first SDG is poverty, when we know that poverty, social inequity is the major driver of the difference in lifespan and health span in the world?
Yeah, that's a great question. And it's crucial and fundamental. The reason that we live longer than we used to is because we have antibiotics, we know to wash our hands, and we have, antibiotics is actually a big one, and we've been able now to cure a lot of age-related diseases, or at least postpone them. People used to die of heart attacks much more than they do now. They still do, but it's later. And so, as a consequence, and also we have food. So even, for poverty, you need to give these people food. This is like the first step. So yes, this is more important. And we heard about it yesterday. And yes, if we could either give enough people food or this, I think I would go for food. I'm hoping that we can do more than that. And so the thing is, though, when you give a person an antibiotic or when you prevent them from dying from a heart attack, you don't make them younger, you keep them alive. So now you have a person alive, and that's a terrific, wonderful thing that they're alive, but they are more frail. So you do end up with a larger segment of the population that needs health care, that really doesn't function very well, depending. Some people do, but many don't. And so that's what I'm talking about, that little segment. I think it's like the planet. There are all these different aspects of planetary decline. I think there are many aspects of human health, and food is one, and this is another.
Thank you. Another question over there.
Thank you. My question goes back to your very final slide. You mentioned that resveratrol, blueberries, and all these goodies cannot be assessed in intervention studies. But I think we have a tool, and this is a retrospective analysis of different groups. You are non-user, and we have very adamant users. So why not push for a study, a large-scale study in the US, let's say via the National Institute of Dietary Supplements and the like, to look at outcomes of different groups, users and non-users, really at a large scale. It still is expensive. It still is a problem that the product in the end is very cheap and consumed in an irregular way. But I think we need to think about creative strategies to address your question.
I agree with you. The problem is, it's tricky. It's tricky for many reasons, and people are doing that. You read about this all the time in the New York Times, for example, or whatever, all newspapers. There's been this study and coffee is good for you, or a little bit of wine is good for you, and then maybe no wine is good for you. A lot of wine we know is not good for you. But the problem with these, they're good. And I really do believe that from all that, I think you can say that the Mediterranean lifestyle and diet is good for you. I really believe that, and that's doing what you said. It's looking at different populations, different lifestyles. We know exercise is good for you. But that's not the same as asking whether you should take resveratrol. It's a small molecule that's found in red wine. You'd need 1,000 bottles to get enough to test what was tested in mice that was not for lifespan, but for health span, it was beneficial. So for that, what you want is a very carefully controlled clinical trial where you just take people and you look very carefully at their lifestyles. What are they doing? You get a very good inventory of their lifestyle. And then all you change is whether or not they're taking the resveratrol pill or a placebo pill, a pill that looks like it but doesn't have anything in it. That's the only thing that you've changed. And then you'll know, should we take resveratrol? And if you do a really good study, it gets complicated, but you might be able to say, well, for this kind of person, maybe it makes no difference. Like maybe if you're pre-diabetic, it makes a big difference, and if you're not, it makes no difference. Who knows? But you can even maybe, with a large enough population, you can retrospectively try to ask which portions might have benefited if you have enough power. So yes, I think your approach is terrific, but it's confounded. Also, the thing is, people who eat blueberries, they're kind of expensive, so chances are they have they have good medical care, they probably exercise, they do a lot of other things. It's really hard to tell if it's the blueberries or something else. Okay.
Cynthia Kenyon, thank you so much.
Thank you all.